All right, so let's go ahead and dive right in. I'm going to share my screen, and we're going to jump right into the content for today. So let's go ahead and start recording. So I'm going to get my recorder on. Make sure we're good to go. All right, here we go. All right, okay, so moving on. So we got a chance to talk a little bit about cancer. And with this question, it kind of warms you up, hopefully, with your mind that, hey, cancer is something that is almost a part of a natural process when it's not caught. And unfortunately, how cells work is that cells have all these functions that we already have talked about. They play a role to acquire nutrients and material to generate energy. They have to be able to interact with its environment. Ultimately, it has to reproduce and adapt. And this cell on the left side of this picture shows you some of these critical organelles. These organelles are the machinery the cells require to perform its function. So you have the nucleus, that's for DNA, you have ribosomes, mitochondria, the lysosomes, the cytoplasm, the Golgi apparatus, but each of these structures have a dedicated role they play for the cell. All right, so we're going to fast forward a few slides here. We covered all this already, and we talked about cell division, and we talked a little bit about the fact that DNA is where all that information that codes for what a cell is going to output is located, right? DNA is inside the nucleus. That's the genetic information that is made out of 4 bases: the guanine, the cytosine, the adenine, and the thymine, right? These bases then generate the ability to pair up and form this helical structure, and that's what you see. So what is the purpose of taking this genetically stored information and then sending that out as a message and then creating protein? Well, this flow that you see on my screen here, this flow from DNA down to RNA down to protein, is basically what they call the central dogma, the central theme of all of biology. Life as we know flows in this direction. It goes from DNA, RNA to protein. There are incidences such as the HIV, and especially with our COVID vaccine technology, that they take RNA and they reverse engineer it so that it goes back into DNA and then goes from DNA down the flow again. So this concept is simple, yet it's very powerful because it can generate different ways of getting a cell to respond, right? So the end goal, whenever you hear about cellular response, whenever you hear about genetic expression, whenever you hear the word getting a cell to do its job, what the cell does is it releases some kind of protein, and that protein will have an effect on your body. So this first step, going from step number 1 to step number 2, so going from 1 to 2, we call that transcription, and then going from 2 to 3, we call that translation. All right, so transcription is the copying of DNA to RNA, and then the copying of RNA to protein is called translation. So just make sure you don't get confused with those two words. They both start with a T, and they all have TION as the suffix. So just keep that in mind, that this is 2 processes. One is from DNA, RNA, and then the other is RNA to protein. So let's take a little closer look at what happens. So the first part of transcription is that within the nucleus, there's an enzyme called the Rna Polymerase. And this is a well-studied, well-documented enzyme, polymerase, which actually will take the bases, begin reading the DNA, and then attaching the correct RNA base to match that. One little catch to this story is that in DNA, we have A pairs to T, and G pairs to C. But in RNA, the T gets replaced with uracil. So instead of T, we have U. So it's AU. So this then changes the way the code is built, right? So you have AU, right over here, as you can see, AU, and then this is AT, because T is still part of the DNA, right? This is part of the top strand is the DNA, right? So that's why you see the T over there. And the bottom strand is RNA, so every time you have a T, it always pairs with the A, but every time you have an A on top, it pairs with the U. So that's why it gets a little tricky when you're trying to write out the code, or if you're trying to read a line of nucleotides, but just keep in mind if it's RNA or DNA. And then eventually, you're going to generate this long strand of mRNA, which stands for the messenger RNA. You can just remember this M, this lowercase letter m here that you see. It just means message. Right? It's the message of the DNA. And now this message is going to be modified, cleaved up, and eventually make its way out of the nucleus. So the nucleus is where all of that magic occurs. All right, so those are some of the key points to keep in your mind as you're doing this module. And this is basically molecular biology. This is molecular biology in a nutshell. All right, and what happens here then is after the DNA is copied to RNA, now you have this information that gets to go into the outer compartments of the nucleus near the cytoplasm and inside these machinery. And the machinery we're going to focus on is the ribosome. Ribosome is a big red globular protein that has the ability to take the mRNA that got generated and start reading those nucleotides and see, hey, what exactly are we trying to build? So there is a rule, or what they call the frame of read, called codons. Codons is 3 nucleotides that will code for a specific amino acid. So this is literally how our life and our cells build everything, right? Build all the proteins based on these amino acids. And you can see right here that obviously this is just an example, 3 different codes. So it's GGU, AGA, ACC, right? So you have 3 sets of codes that will then correspond to the bottom part, which is going to be bound to, if you can see right here, there's a little round T-shaped, almost like a little cross-shaped protein, little structure that has a round amino acid right here, right? These round little circles are amino acids, glycine, arginine, threonine. These are different amino acids that have different properties. And depending on what code it is, it's going to match the anti-code to it, right? So when I say anti-code, I mean this. So if you look at this first line, I'm going to use another marker here so it doesn't confuse you. So I'm going to use red. I'm going to highlight this part. So if you look at this upper part of this transcript, you have GGU, AGA, and ACC. So these are going to be the mRNA information. So the mRNA information is going to be read by the ribosome, and they're going to then take the corresponding base paired anticodon. So that's going to be G paired with C, G paired with C, A paired with U. So CCA, that brings glycine. So let me go back to what we just talked about with cancer, right? So you just talked about the fact that cancer is a mutation. Cancer is when cells divide uncontrollably, all that bad stuff. So when you think about your cells having a mutation, what that really means is that some step along this process of transcription or translation, something goes wrong. All right, and so let's say in a healthy cell, we need GGU, AGA, ACC. This is the 3 sets of codes that we want. But because you were exposed to a nuclear bomb or radiation, or maybe you ate some chemicals that got into your system, that toxin, that carcinogen, caused the ribosome or caused your DNA to get damaged. And when I say damage, what I mean by that is that it made this T right here into an A. So now instead of normally you should have an A over here, you have a T, right? Because the A pairs with T. And now because you have a T over here, down in this bottom chain, there's a T, and T then pairs with another U. Okay? And because there's this mess up of the base pairing, so let me write this out here. So 1 second, I need to use a smaller line here because it gets on, right? So A to T, right? G to C. That's going to be DNA. And then for the RNA, you have A to U and G to C. Okay? So imagine you mess up this pairing, you mess up the bases, then what amino acid that is going to bring into this protein will be different. And so you could have a risk of creating a protein that's non-functional because of some mutation either at the DNA level or at the RNA level. And that's really what it comes down to. Usually when you have like one mutation or one mismatched base pair, it's not going to be the end of the world. But when you have a series of bases that are completely off, then you completely throw off the protein that gets generated and you nullify the function. So what's the deal with protein, right? So the point to make here is that proteins have a specific shape, and when you disturb the change in shape, let's say you accidentally change a few amino acids and you change a few bases, now this amino acid is supposed to have a certain kink, bend, property, or shape that you want it to have for its function. There are 4 levels of organization or function to the way that proteins work. The first level is called primary. Primary just means that you have a dedicated, specific kind of amino acid that outlines that entire chain. Right? So, for example, you might have the very first amino acid as valine, and the very last amino acid as glutamine. You have a certain set that you have to abide by. And if you don't have that set, then there's an issue. The second level, or the second structure, secondary structure, is going to be when you have a specific bend as well as a kink or a twist to that gigantic set of amino acids. And there's 2 structures that we talk about, one called the alpha helix, and the other one is called the beta sheet. And these are just simply bends and kinks that occur because of specific amino acid residues. Some of these might have a little charge on them, some might have a little bit of a positive and negative ionic interaction, and as a result of that, there could be a twist or turn that occurs. The third level is called the tertiary, and that's when you have a 3-dimensional view of an actual functional protein. So this is where things get exciting, because if you disturb this 3D shape, then the function might not work anymore, right? So imagine you had a key, and that key is completely bent and damaged and deformed, then it might not be able to fit properly into that keyhole. And then the last tier here is going to be called the quaternary, which is going to be when you have multiple proteins. As you can see on the picture, you have a green protein, you got a red protein, you got a purple protein, and they're all interacting in the right way. Right? So if you were to have a mutation and the protein is supposed to be, the green one supposed to be in the back, is now in the front, then that messes up the normal interaction it's supposed to have, and that's going to cause problems. So that's the way you can visualize and think about protein, think about amino acid, think about DNA, think about cancer. So I have another question for everybody for the day. So this question is a multiple choice one, so you can just respond at your own cell phone or own keyboard. So the information for producing a protein is contained in which part of the DNA molecule? All right. So is there a specific part of that DNA molecule that codes for what the protein becomes? So you need to think about what is the different parts of a DNA molecule. And of course, we talked about these bases, right? We talked about the deoxy sugars, and we talked a little bit about the phosphate linkage that glues them together. All right, so I'll give you guys 2 minutes. If you need a little bit more time, I can just raise your hand if you need a little more time, and I'll add that. All right, so go ahead and respond to this question. This is the one for the day, the new question of the day. All right. All right, so I got about 40 people responding, so another minute or so. So go ahead and key in your response. So another 10 seconds here, and then we will move on to the next question. So as we're going through these questions, you're probably thinking, Lynn, what are we doing? Why are we doing these questions during our session? Well, these questions are practice questions for you for the exam, right? The whole point is a lot of times you take a STEM class or biology class, and when you take the exams, you're like, oh my god, the questions are confusing, I don't know the answer. A lot of it is really just vocabulary, so you just gotta read the book, read our PowerPoints, read the slides, make sure to write down, highlight, circle the words you don't know, define them. That's a start. And of course, as you're going through the question, make sure to answer them the best you can so you learn in real time from our Zoom session. All right. I see 79 people out of 84 answered. So if you haven't answered, please answer the question, because you get points, right? And these are easy points to earn. So you get them regardless of getting it right or wrong. And that way you hopefully get it right on the exam. All right. So, pretty even spread here. So we got 28% saying A, and then 14% saying B, 34% saying the phosphate, and then 24% saying D. All right, so the answer is actually the bases, right? The bases that have the A, the T, the G, the C, those bases that generate the interaction and linkage is what determines really the information for coding. Okay, so this one is a tricky one, and you can see, if this was on an exam, we would have, only had a few people get it, only a fraction of the class getting it right. So the answer is A. All right, let's do one more here. So what molecule helps to move genetic information from the nucleus of the cell to the ribosome? So this is a machinery question. So we're moving that information, right, the genetic instruction now into message, and then the message into the ribosome for the protein. So which one of these is what moves that information from the nucleus to the ribosome? All right, so hopefully this one is a little easier. We hit on this one today. All right, and then I will be right back, folks, and I will give you guys a chance to answer the question. All right, so another 20 seconds here. Another 20 seconds. Go ahead and put your answers in. Yeah, Jasmine, you good? You got the chance to answer the question? Everybody good? All right. Cool. Here we go. Okay. All right, so let's take a look at the answer. So the answer for this next one is mRNA. Awesome, good. So the answer is mRNA. The messenger RNA is the molecule that transfers the genetic information as DNA into a message form so that it gets to go to the ribosome. That's good. Awesome, guys. Great job. So those are the 2 questions for the day. So let's go ahead and finish a few more slides here. So the next phase of this discussion is now that we talked about the ribosome, now that we talked about the transcription, translation, and the point that DNA serves, what's the end result? The end result here is that cells have a life cycle. Just like most of us, we have different phases of our life. We go to college, we graduate, and maybe go to grad school, or maybe get a job. And then after you get a job, you want to save up and eventually maybe get married, or maybe buy a house, whatever you want to do. There's different phases of your life. As far as cells goes, they also have a couple of key landmarks in their cycle. So we start off with what they call G0. G0 is when the cells are not doing anything. They're stagnant. We have this gigantic phase called the interphase. This is when the cells are pretty much in a prep phase to get ready for this very big event called the M phase. And the M phase is the famous one-word phase called mitosis, which is none other than nuclear division. And you'll see in this picture that there's a few more small phases in here that might be a little bit elusive, but you'll see there's one that says G1, and there's one that says G2, right? So what does G stand for? Well, one way to simplify this is G stands for the word growth. So when it's in G0, we call that growth 0, otherwise known as not growing, not doing anything. So that's when cells are pretty much done dividing, they're stagnant. Some examples of these cells are like your heart cells, all right, the cardiovascular cells in your body, some cells inside your brain, like some of the neurons in your brain. These cells generally are finite. So that's why when you hear about Alzheimer's or as you age and these very nasty neurodegenerative diseases that will consume and just decay down the neurons, it's so sad because we only have so many neurons in our brain, and as you lose those neurons, then you lose the brain's cognitive function. G1 are cells that are getting ready to divide, so they're getting larger, they grow in size, their organelles double. And then S phase is when you have DNA replication. That's when DNA copies itself and the chromosomes duplicate. And then G2 is a phase right before the splitting of a cell. All right, and so we're going to focus our discussion into mitosis, which is the M phase. And you can see in this picture here that the M phase is divided into roughly about 4 to 5 phases. Technically, it's only 4 phases, but that last phase that you see, called cytokinesis, is just a phenomenon of dividing the cytoplasm. All right, so we really have 4 phases: prophase, metaphase, anaphase, and telophase. And cytokinesis is just a part of that final cell division. All right, so that's the storyline that we're going to get into. And just to warm you up, you'll see that obviously cells have the opportunity to call it quits and to call and to really catch any errors. There are really only 3 checkpoints in a cell's life cycle, and if you miss all 3 of them, then that means that cell or that DNA has the ability to be mutated and then evade the consequences and potentially become cancerous. So if you look at this picture, I want to ask you guys, we're not going to answer it today, but just think about which 3 places on this screen you see is where it causes it to stop and serve as a checkpoint, and also why. All right, so the first checkpoint you can see right here is what they call G1 checkpoint. This is right about halfway in to G1. And then the second checkpoint is at the end of G2. And then the last checkpoint is going to be during mitosis, but more specifically in the metaphase, anaphase phase, right? So you guys can see that there's 3 of these red blocks here. And those are the stop signs. Those are the opportunity for the cells to completely reset itself or to destroy itself if there were to be an error.